This is really not working. Oh, hold on, I'll get that in a second. Trying to get this started. A camera and therefore a microphone that this computer uses is like super touchy. So sometimes I just freaking turn the screen, it like plugging like while I'm touching the wire. So I'm trying to get that set up really quick. Okay, microphone good. Video on. Great angle. You get the double chin going. What? No. Just enough. Just enough so that you just don't forget that it's there. Okay. Yeah. Yeah. You inspired me. I might do a song for one of mine that I'm doing as an example for you guys. It's not going to be, don't expect like something great, but. Okay, it's just like this is what it did the other day. Oh yeah, and it just did that. Yeah, I don't know why it does that to some computers. I just hope it doesn't do that to you when we do the exams, is what. No, that's just from when I have a list of literally everybody in alphabetical order, and I roll the dice and I'll write the numbers up and then I go find the names. Yeah, that's all that is. Oh yeah, oh thanks. I'm not gonna turn it down for sure. All right, let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So I still got 3 people that aren't signed in quite yet, or it's not registering that you're signed in. We're gonna get going here in a second. I don't want to wait too much longer. I know there are people that are out too. I don't know if you guys have been paying attention, but the flu is going around, like really going around. I don't mean like a person has it. I mean like half the freaking state has it. So yeah. So that reminded me actually, I'm not sick and I haven't had any symptoms and I have had no fever, nothing. And I've been testing because my husband has flu. So I've been trying to remember to mask up. I forgot I take it off when I drive my car, but I don't have it and I shouldn't. I figured for how long, seriously, how long my husband's had it now, it's been like 4 days. By now I would probably have tested positive. It doesn't take very long to show symptoms from the flu. It usually only takes 1 or 2 days. So it shows up pretty quickly, but just be safe. But to be aware of that, if you have symptoms or somebody that you live with has symptoms, just be mindful of that. Because he got it from his coworkers who like came to work with a fever and everything, and then he's like, no, but he still got it. So I think it's just a strain that I've had before. Okay. We're just about to get started. As long as you get a question in, you'll be fine. So I'm gonna go ahead and start now. And remember, if you guys come in late, that code to sign in will be the bottom of the screen. If you can't read it, just holler, I'll tell you what it is. What type of chemical bonds involve sharing electrons? Start off with an easier question. Most of us know this one. Yeah, the questions are over the previous chapter, so that's chapter 2. Anyways, so yes, so this is covalent, Oh, yeah. Sorry. Yes, so covalent, you guys, most of us got this one. So this is, of course, going to be like cooperation, working together. All right, this question's been getting people. Be sure you're reading your choices and understanding them when you choose them. Which one would match up best with this? What would be the best representation if you had to choose one of these choices? All right, so you guys did do better than the other sections, believe it or not. So the answer for this one is going to be the green one, holding water molecules to one another. We know that the polarity of the oxygens being negative and the polarity of the hydrogens being positive, those attractions are holding the actual water molecules near one another, and it's just attractions. So if we look at the blue one, which is also the more popular one in the other sections, and like a majority of the other sections, we're choosing that one. I know. So holding 2 hydrogen atoms together in a molecule of hydrogen gas, here we would be talking about like this sort of chemical equation where you have H2. That would be hydrogen gas, right? In that situation, you have a H that is bonded with a H. What type of bond is this? Do you guys know? It's not a hydrogen bond. It's a covalent bond. Okay, so those guys are actually sharing their electrons in that case. So it's not this one because that would actually be a covalent bond. Most of us know that they're not really strong bonds. Our hydrogen bonds are actually just attractions. So they're pretty weak bonds. And then as far as the yellow one goes, between hydrogen atoms in different molecules, that was another popular choice in my other sections. But the reason that one is wrong is because in that case, we would be talking about if we had a molecule like water, where you have your O and you have your H's, whatever. And then these H's have their little positive polarities. Now you're talking about if we had another molecule out here that had an H associated with it, and maybe it's attaching to an O, and that O would be negatively whatever, doing something else, and this would be positive, and that'd be doing something else. That these guys here would be attracted to each other. And that's not going to happen because those are the same polarities. So positive is not going to be attracted to positive. They're always opposites that are attracting. Also, when we're talking about hydrogens having polarity, it's always going to be positive polarity, okay? They're not ever going to hog the electron and become negative polarity. So in order for opposites to attract in our hydrogen bonds, the hydrogen will be positive and it'll be attracted to something else, usually negatively, usually it's oxygens, okay? So remember that, and that should keep you straight because this is not what's happening. That help a little bit as to why the green is the answer. Now, is that the definition of a hydrogen bond? No, but it is the only one that applies to hydrogen bonds being represented from our choices here. So this is a pretty good one of those examples of being aware of, if I'm asking you, and if it was a test question, I'd put more words than just hydrogen bond, right? I'd probably say, which of the following would be the, from the following choices, which would be the best representation of hydrogen bond? And then you would have a little more leeway into that. But just be careful you're not looking for definitions all the time, because sometimes that can be what like you're trying to look for what looks most like the definition, and you get tripped up on this. We know that bonds that hold water molecules to one another, you might be aware at least that they are hydrogen bonds, but you know that's not the definition of a hydrogen bond. And some of us are going to steer away from that answer because of that. But none of the other ones are hydrogen bonds either. So that's where we have to reassess our choices and just be aware of I forgot that it does this. Be aware of the content. All right. That's how I feel too. Okay, so question 3, I promise the other ones aren't that bad. What element is in all organic molecules has to have at least this. Most of you guys know this one. For some reason, it's only giving 10 seconds on these ones. So don't beat yourself up if it's not, like, you're not getting an answer in. So carbon, you have to have carbon. And then that carbon has to be bound to hydrogen somewhere in order for it to be considered truly organic. But that carbon is really what's delineating it, right? Yeah. All right, true or false? Water is a poor solvent. It's a bad, not P-O-U-R, but P-O-O-R. Is it bad at being a solvent? All right, so this one's pretty false. And in fact, we know this has to be true because it's the solvent of life. So we depend on the fact that it's good at being a solvent, especially for polar molecules. All right, next question. What is water's pH? I know most of you knows this one. Good job, guys. So we got that down. It's not something you have to worry about. All righty. Next question. What range on the pH scale would acid be? Sure you're reading your choices. Yeah, there it is. There it is. Yeah, sorry. So yeah, 0 to 6.9. If it's less than 7, it's an acid. All right, that's the problem with having timers on these two. All right, lipids. Another one where I'm giving you a word. Yeah, so this is gonna be all of the above for these. They can be used for energy. We know this because that's why your body stores fat, so it can break it down later for energy. We can use them to make hormones. We talked about the sterols for that. And obviously the cell membrane, where we have our phospholipids, which I think is a question on here. Which of the following organic molecules is amphipathic? Go figure. All right. So our amphipathic molecule, the one that we specifically delineated as being amphipathic, was phospholipid. So if you didn't remember that we specifically delineated, we could go through the other choices and talk about why they're not. Glucose, it's a sugar. Nobody picked it, but if you had been considering it, sugars. If you take sugar and you put a little bit in your water, does it dissolve? Probably polar, right? So we could probably eliminate it just based on just that theory. But sugars, a lot of time, like our basic one, like glucose, its overall chemical equation would be C6H12O6. It's got a lot of oxygens in it, so it's probably going to have polarity to it, okay? Next one, amino acids, which is the one that some other people chose. Amino acids, they've got the amino group, and that has a positive charge to it. And then we've got the acid end of it, that COOH, carboxylic acid. That group tends to have a negative charge because of those oxygens. Now that's not what makes it amphipathic. So amphipathic is always going to be talking about charge and then hydrophobic. So if it has that polar and it has a hydrophobic aspect, both. So can amino acids have hydrophobic R groups? They can, but do all of them? No. So some of those R groups can have charges or polarities, and some of them are non-polar and can be hydrophobic, but not all of them. So we don't want to group all of them, but all phospholipids have that phosphate with a negative head and the lipid part that has the hydrocarbon hydrophobic tail, right? For nucleotides, nobody picked that one, and it's going to be, I think, a question on here. But the nucleotide consists of the sugar, which is a 5-carbon, right? And we already talked about sugars having polarity. And then we have the phosphate group, which we know has a charge to it. We already talked about how many oxygens are in that, right? And then we also have a nitrogenous base. So that's what makes it A, T, C, or G. Now, if you weren't sure, just off the top of your head, if that part had polarity or if that was hydrophobic or something, think back to when we talk about DNA. We have DNA, our sides of our ladder is just repeating the sugar phosphate, sugar phosphate, sugar phosphate. In the middle of the rungs of the ladder, the thing that holds the two sides together, that's our bases, our nitrogenous bases. And their attractions to one another, you remember how they are attracted to each other, what bond that is? Hydrogen bonds, right? So if there's hydrogen bonds between those bases that's holding your ladder together, that has to have polarity because you've got positive hydrogens being attracted to negative oxygens. So there's always oxygens that are attracted to. Does that make sense? So you could also eliminate that out just based on your understanding of the DNA ladder, telling you how those bases work. Even though we haven't gotten too much detail on those yet, we will in chapter 6, but that deductive reasoning. We just talked about it, but what is the monomer of a nucleic acid? Thing I was just talking about. Gosh, it's hard to breathe with this on. I don't even know why. It's far enough away. I'm not going to worry about it. So this is the nucleotide. That's actual monomer of any of our nucleic acids. So DNA, deoxyribonucleic acid. RNA, deoxyribonucleic acids. Those are nucleic acids. Those building blocks of those guys are the nucleotides. Even ATP, that energy molecule we talked about, is technically a nucleotide just with extra phosphates on it. So, right. We're getting it. Last question. Alpha helices, and it's supposed to be helices, and beta pleated sheets are examples of which type of protein structure, or which level of protein structure? Hands are so dry from this dry weather. Okay, so this is the primary. No, sorry, secondary. I don't know why I said primary. I was like looking right at primary and I don't know why I said it, but yeah, it's secondary, excuse me. So primary is the amino acid sequence. That's just the amino acid sequence, like they're spelling it out. Secondary, we have those R groups that are on each of those amino acids that make each one different. The ones immediately next to each other, causing it to start making those curls that we see for the alpha helices, or wanting to stay straight, like we'll see straightaways just going back and forth for our beta pleated sheets. Then tertiary is like how that whole structure is going to start reacting with the rest of the structure and getting our 3D shape in there. So that's the tertiary. And then having multiple of those clumps come together and work together to make the functional protein. That's the quaternary. Okay. Yay. Good job, guys. Will our podium come up? Again, it's okay if you didn't do well on it. It's participation points, but start getting us thinking about how we should be understanding the material. You haven't had a chance to look over the critical thinking questions. They're going to make more sense when we get through chapters 4, 5, and 7. Understanding the differences between the things we talk about in 4, 5, and 7 is what's really going to help you understand some of those questions. Some of them you might be able to answer after chapter 4. But the whole point of those, I think when we get to breaking them down and talking about like how to answer those questions, because it's easy enough for me to give you the questions and give you the answers. But understanding why the wrong answers are wrong and why the right answers are right, because those exact questions are not going to be on your test, but it's helping you understand how to assess those questions. Anyways, yeah, so when we talk about breaking those down, you will realize if you understand the material well, that what those questions are really asking are a lot simpler than probably what you think. It gives you a lot of information, but probably what it might just be asking is, which grouping does this organism fall into when you break some of these down? So just be able to read those questions and figure out what the question is really asking and break down what information you need from that question to answer that. Somehow the camera is still working. It's like a miracle. Okay. All right, so let's get out of this and then we'll pull up our slides. We'll get to chapter 4, where we're going to be talking about bacteria and archaea, mostly bacteria. We'll touch on archaea a little bit. All right, so bacteria and archaea, these guys are eukaryotes or prokaryotes, same interchangeable concept, but what those guys lack is a nucleus. And so nucleus is a membrane-bound organelle whose function is just to contain your genetic information. These guys don't have a nucleus, so their genetic information is just hanging out in their cell goo. It hangs out in a region called the nucleoid region, and it kind of tightens itself up and winds itself up into that area. So it's not making a big old mess everywhere in the cell, but it's not contained in a membrane. Nothing is, except for the cell itself. So these guys have an outer membrane that contains the goo inside, which we call the cytoplasm, but that's the only membranes that they have, in addition to maybe an outer membrane that surrounds the outside. We'll get into that though, but no internal structures. Eukaryotes, on the other hand, when we get in chapter 5, you'll see that they have all these membrane-bound structures, including a nucleus, the membrane-bound structure that contains the genetic information, the endoplasmic reticulum, the Golgi apparatus, the mitochondria, chloroplasts, all those guys are membrane-bound, and they have little functions that they perform for the cell. Just compartmentalized. This is a good kind of study reference for the structures that you have to know. Go find the ones that we would have to know, and then it would show you where it is on the picture of the cell. We're going to go over these kind of individually as we go through. So bacteria and archaea, both of them are single-celled organisms. They're never going to be multicellular. They're always single cell. And each of these cells is capable of reproducing. They each have their own metabolism. And metabolism, what really metabolism is all the chemical reactions that go on in your body, all of them. Whether we're making new molecules or breaking them down to use them for energy or parts to build new things, the whole thing is your metabolism. So each cell has the ability to reproduce, has metabolism, and the ability to process any of those nutrients that it gets from breaking down things that it takes into the cell to use as components for building new parts and creating energy for the cell. Bacteria can act as a group, but they don't depend on that. Like our cells actually depend on acting together for a functional organism. If I take one of your cells off of your body, it's not going to grow into a whole new you given time, right? But if you take a bacterial cell away, it can grow into a whole new colony all by itself. It can make a whole new cell that can itself continue to grow a whole new colony, and so on and so forth. And it's the kind of the whole premise of how we're going to be dealing with them in the lab. So they can act as a group in colonies. They can also work in what we call biofilms, which are kind of like when we get layers of different kind of species of bacteria all living together. And what you would imagine what a biofilm sounds like, sort of a sticky old film, like a slime layer or something. We have different kinds of microbes all living together in this layer. And then nanowires, which we're not going to talk about in this class, but it's something that exists. This shape of our bacterial cells. So this is just talking about bacteria, not necessarily about eukaryotic cells, okay? For bacteria, there's 3 general shapes: the coccus, the bacillus, and curved. And I'm not really going to test you guys on curved too much, but when we start talking about spirochetes or spiral-shaped cells, they're obviously not cocci or bacilli, so they fall into that curved category. But the coccus, we see, this is how you can remember the difference, okay? C-O-C-C, those are very round letters. Coccus is the round or spherical shaped guys. And then the bacillus are our rods. So you see the L's in bacillus, those look like the rods of bacilli. Multiple for bacillus, singular. So cocci versus bacilli. Round versus, says blocky, we're talking rods really. Okay, I'm gonna show you some pictures. And when I test you guys over this on the exam for the lecture portion, I'm going to talk about more like definitions or explaining, like you see in the microscope some rod-shaped cells. How would you call that shape? Bacillus, okay? On the lab exams, you will be given a microscope that has a slide on it, and I'm going to ask you what shape was it. So when I do that on the lab exams, I'm going to give you obvious examples, okay? Because these guys, there's gray area. Obviously there is. In fact, may or may not use this organism, but we do have an organism that I sometimes use in the lab. That is a coccobacillus. And most people are gonna be like, is your organism a coccus or a bacillus? And so poor old group that ends up with coccobacillus, you'll have people that are arguing, I think it's coccus and I think it's bacillus. And I'll come over and I would tell your group that I would be nice to you. And I'd be like, you have a coccobacillus. Sorry. But I'm gonna try not to use that one if I can help it. It just depends on what I can get to grow. They don't always behave. They're living organisms, and sometimes the dryness of the environment or the humidity, whether or not my incubators are going to behave, and like our water. Over the summer semester, I found that our water was pH 3 out of the tap. So things aren't going to like that. That's not real happy for life, right? And so when we found out, that was out of our deionized water tap spigots. So we're talking about like treated water, So we've gotten that issue fixed, but I was having a big issue with things growing, and I thought it was me, but it turns out it wasn't me. So I felt a little better about that. But I've been growing bacteria forever. You think it'd be easy to grow bacteria when you want it to grow, but it wasn't behaving. Now we know why. So definitely know the difference between coccus and bacillus. Those are like our biggies. And I only have cocci and bacilli in the lab. I don't have any other shapes. So it's the cell shape, all right? You can also have pleomorphic cells. We won't see those in the lab, but they do exist. We'll talk about why they are pleomorphic in a little while, but pleo means many, morph means shape. So these are just guys that have random shapes. They don't have a set, they're not hooked onto coccus or bacillus. They can kind of be just irregularly shaped. There's a reason for that that we'll come to later. But here's some pretty good examples of some shapes and arrangements. We're going to talk about the arrangements in a little bit. But here we have the cocci. And like I said, when I test you guys over this, it's going to be round versus rod. And you are going to be able to tell, okay? So you can see in these images here where we have this staphylococcus and this streptococcus, it's in their names, for crying out loud, right? So Staphylococcus aureus tells you that the cells are coccus, round. Streptococcus pyogenes, it tells you coccus, round cells. Now you can already see the differences between the arrangements between these two. And that's what the staphylo and the strepto mean in these names. Not all cell names, obviously species names aren't always going to give it away. Obviously Legionella pneumophila isn't telling you anything that it's a bacillus. Escherichia coli, it's a bacillus, it's not in the name. But some organisms will give it away in the name. If it says streptococcus, that means it has a strepto arrangement, and we'll get to the definition of that in a moment. And it tells you that the cell shape is coccus. It's not going to lie to you. You're never going to have a streptococcus and I'm like, actually, it's bacillus shaped. No. So if it says it in the name, you can bank on it applying. Okay. So we have our cocci here. You can see that these guys are spherical shaped. These top ones, the staphylococci, they're irregular bunches. That's what staphylo means. These guys down here, the streptococci, that's what strepto means. So I feel like strepto, you could almost say it sounds like strip. So it's like strips of those round cells. That's how I remember it. Strips, they say chains. I feel like strip and strep sound so similar, whereas staphylo, you just have to remember that that's going to be irregular or grape-like clusters, okay? Then we have our rods. You can clearly see the difference here between our examples that were given, right? And you can see these guys are rods. Don't worry too much about the myces. They kind of branch off each other really weird, and we won't have to worry about them. Like I said, when I give you guys examples, it'll be really obvious, like these guys versus these guys. Pretty obvious here. Our curved ones, you can see these kind of have a comma shape. I would never give you this on a test and be like, is it bacillus or is it coccus? And actually it's a vibrio. Like, forget it. I'm not going to do that. So I'm going to leave them obvious for you guys. But the spirochetes and the spiral shaped guys, they're pretty obviously spiral shaped when you look at them. So anyways, that's the shapes. So now we're going to get into the arrangements. And there's arrangements that cocci fall into, and then arrangements of the bacilli. There's a little bit of crossover. So obviously with diplococci, we're talking about pairs of cocci. So pairs of round cells. Diplo always means two. A really, really good example about this one, and one that's going to apply really well for our gram stain when we're looking at what the gram stain is and why it's important and everything, is Neisseria. So Neisseria is a genera or a genus of bacteria, and there's a couple of species of Neisseria that are pretty medically important. So Neisseria gonorrhea, and then Neisseria meningitidis. And so we more commonly refer to Neisseria meningitidis as the meningococcus. And you've probably heard of it as meningococcal meningitis, right? So they'll warn you about that meningococcal meningitis and how serious it can be, especially if you're living in like dorm situations and things like that. So it is a diplococcus, very characteristically so. All of the Neisseria are. And they're also gram negative, which is also going to be important. We'll talk about what that means in a moment. But you could even just do a simple stain of these guys and you could see those diplococci. You don't need to even do the gram stain. And those diplococci are going to be so pronounced that the most common cause of bacterial meningitis is not Neisseria meningitidis. That's probably the second most common. And it's the worst to have. But the most common, which is the second worst to have, is going to be Streptococcus pneumoniae. Sounds like it causes pneumonia because it does. It's also the most common cause of pneumonia. So we're talking about something that can actually colonize you normally, but when it gets into the wrong place or it grows in too much of a number, or if you don't normally have it in your body and it's produced from somebody else, or if it's antibiotic resistant, all these things that can throw things off, then it can become pneumonia or it can become meningitis or whatever, okay? So Streptococcus pneumoniae, also known as the pneumococcus. So pneumococcal pneumonia versus meningococcal pneumonia. Streptococcus is gram positive. So you could tell the difference with the gram stain there. But a diplococcus, which just pairs, they look like butts, like legitimately, versus a streptococcus in chains, you would be able to tell the difference with a simple stain with that. So already we're starting to get the importance of looking at these guys. You don't even need to culture this. You can just get somebody's CSF from a spinal tap and do a stain on it directly, and it'll tell you that information. Because spoiler alert, your CSF naturally should be completely sterile. It should have nothing in it. It shouldn't even have white cells in it. It should be nothing, okay? So those are gonna be giveaways when we're starting to look at the importance of understanding this stuff in the perspective of the medical relevancy here. So diplococci, already learned a thing. Tetrads, tetra 4, okay? Staphylo, irregular clusters, not ever gonna ask you about micrococci. Streptococci, chains or strips. And sarcina is sort of like these weird cuboid arrangements. Never gonna see it. All right. The bacilli are rods. They can also be in pairs, so they'll just be end to end, right? Streptobacilli, strip of bacilli, so they're just gonna be long chains lengthwise. And then we have this other term, palisades. So you may have heard the term palisade referring architecturally, or like, I guess, what do they call it? Civil engineering, where palisades is referring to barriers and like a fence-like barrier. So a palisade is like fences. So these are like fence posts. So these guys are like upright, vertically next to each other, literally just like a fence. That's a palisade arrangement. So if they're like this lengthwise, that's going to be strepto. If they're like this next to each other, palisades. So only bacilli can do palisades, obviously. All right. The spiral shaped guys can be in chains, or they can also kind of be arranged next to each other. I'm never going to ask you about them. So definitely know your cocci and your bacilli. Okay, so we'll ask you about those. Next, we're getting into biofilms. Like I said, it's kind of what it sounds like. It's a film made of biomass. All right. This could be bacteria, protozoa, fungi, and all sorts of stuff that can just kind of coexist in these sticky layers with one another, right? And so it's like a little mini ecosystem, and they often will benefit one another being this way. So they can give protection to one another, food to one another, and help keep each other like aware of what their environments, how they're doing, and stuff like that. So it works to their advantage. The way this works is they find a surface. Usually there's roughness to it that allows it to stick initially. Then they get a sticky coating that allows them to stick better. They make even more of it to help stick together, and this stickiness helps other things stick to them, and they might make even more stickiness, and then within that, all these different species can start to coexist. They can send signals to each other in the form of different chemicals, could be protein signals, could be waste products, and based on whatever they're perceiving from their neighbors, they could decide if that is a situation they want to stay in or if they want to deviate from that. You can imagine that if you went to a party, maybe your kind of party, you might see people, you might want to have a good time and have some drinks and whatever, and you might see people, whole bunch of people there, and there's drinks everywhere and there's food everywhere, and think it's a good time. You go into another kind of party, and it's super dimly lit, and there's a whole bunch of cigarette smoke, and it just is a bad deal. You might not want to stay in that. So you're reading your room that way, and they're reading their room with their own like signals. Okay. So that situation where they can respond to those sort of signals between those different species involved in that, that's called quorum sensing. And just like we would have like in our own Senate or something, you have to have a certain percentage of people show up to take a vote or whatever. That's a quorum. It's a group of people, right? It's the same thing here, group of bacteria and other microorganisms. Best example I can give you of a biofilm would be the plaque on your teeth. It's a lot of different organisms, that's normal inhabitants of your mouth. They're going to stick on the surfaces of your teeth and then have a good old time because of all the stuff that you're eating, sticking on your teeth, and the sugars that are going to be lingering behind from what you've eaten or drank or whatever it is, and they're feeding off of that. A lot of these guys can ferment, and we know that fermentation, a product of that can be acid, and that the acid that you can make from fermentation, we can use that to make kimchi and yogurt and things like sauerkraut and whatever, but they're doing that to your teeth. And the acid that they're making on that is eating through your enamel of your teeth. And that's dental caries or tooth decay. And that's why that's the most common of the infectious, diseases, you could say, of humans. It's the most common one because, people don't floss very well. They can hide up under your gum line. This doesn't make you want to brush your teeth right now. Moving on. So they don't just use that sugary coating, which we're going to talk about that sticky coating in a little bit. They can also use other things to attach to things, and they also have appendages they can use for moving. We're going to talk about them individually. Start with the flagella. The flagella, which is multiple, flagellum is singular. This is the appendage that they use for movement. Not all bacteria are modal. So if you are non-modal, if you can't move around, you don't have a flagella. So not all of them have this. Okay. Staphylococcus aureus does not have flagella. It's non-modal. It's a good example. Whereas E. coli, it makes sense that it would be modal, especially if I were to tell you the following. And we're going to talk about the Fimbriae and the importance of that in E. coli in a moment. But E. coli can travel around in your gut environment, but also in other areas of your body as well. E. coli, we'll get into in a moment, but so it has flagella and actually has multiple kind of. So flagella, what they are, it's like made of protein mostly. And you see how it looks like a little motor that's kind of situated in the cell like envelope, the different layers of the outside. It really does look like that. If we look at it in an electron micrograph, where we can look at things at the atomic level, it's pretty cool looking. But what this does is it causes that protein appendage to rotate. It does not move in a whip-like fashion. They can't control it that way anyways. So the way they control it is by rotating it clockwise or counterclockwise. Okay. That's how they're going to control it. And they can only do that. So how is that going to help them move in directions? We'll talk about it. But first, we'll talk about the arrangements of these guys. So you might have one flagellum, and then you would be monotrichous. Trick, the T-R-I-C-H part, that means hair. So monotrichous, you have one hair-like appendage. This is your flagellum, okay? You can have lophotrichous arrangement. That means you have tuft at one or both ends, tufts of them. So that's image C. That would be our lophotrichous. Image A is our monotrichous. So amphitrichous, where you have one flagellum at each end. So far, these are all polar arrangements, right? We're talking about at one end or both ends. Peritrichous, now we're amping it up. Peri means it's all over it. And so when you have them all over, like we see in B over here, we have them all over. This guy's very, very obviously just coated in flagella. And so that's peritrichous. Good example of that. All right. So these guys are responding to signals in their environment. The two types of signals they could potentially respond to will be whether there's light. If you are a photosynthetic organism like cyanobacteria, the ones that developed our oxygenated atmosphere for us back billions of years ago, those guys are going to respond to light. They might want to move in more closer to where the light is. The other guys generally are going to respond to chemical signals. And you would respond to chemical signals too. And I'm not necessarily talking about pheromones. What I'm talking about here is going to be if I put a freshly, this is always my favorite example, freshly baked plate of cookies, and everybody could smell it. That's chemicals hitting your, olfactory sensors in your nose, and you're reacting to those chemicals. You might want to move toward it. So that's a positive chemotaxis. If I put a fresh plate of dog poop up here, you're probably going to want to move away from that. So chemicals, again, hitting your olfactory, and then negative chemotaxis, away from it. These guys can sense chemicals in their environment. If it's nutrients, they might want to move towards it. And then if it's like toxins or things that make them sense that things are dying over there, they might not want to move towards that. So that would be negative chemotaxis there. So we've got those situations. Chemotaxis drives not just bacteria, but even our own cells, like our white cells, are responding a lot of times to chemical signals being given off by other cells in your body. So that's still chemotaxis. All right, so how do they use their flagella for moving? You can either run or tumble, and that's it, okay? So if you're just spinning your flagellum in one direction, you're gonna run. It's just gonna keep you going straight. So think of it being in drive, you're driving straight. Now, these guys can then go to a tumble, and that's just where they switch the direction. So you're going counterclockwise, and you just suddenly switch to clockwise. It causes them to tumble and change direction. If you pop your manual transmission straight into reverse, good luck with that, okay? But like you're in some kind of drive, and if you were popping in reverse, and the cars wouldn't just freaking stall on you on that, and if it suddenly start going that way, you could see it kind of like almost want to shift directions if the tires weren't keeping it straight on the road. And that's really what these guys are basing that on. It's almost like a random, like reorient and now start going again straight. And then just keep tumbling until you start seeing more cookie smell. And then, oh, there's more here. You tumble again. Oh, there's even more. Or you tumble again. Oh, there's less. Okay, we want to tumble again. You try to see if you can go back that way. And so they'll just keep tumbling and then hope. And then as they're running, if they start seeing a decrease in the thing that they want, tumble again so they can go back in the good direction. Or if there's an increase in the thing they hate, tumble again so they can go away from it. And so that's really how they do it. It's sort of random like that. And what it looks like is this. So we have here, they'll just run and tumble just to investigate their environment randomly. Maybe there's something good over here. So they'll just run and tumble just to get a sense of their environment. Once they sense something they want to move towards or away from, now we're going to be a little bit more controlled in how we're running and tumbling. So we run for a little bit. We start to realize that we're having a little bit of a decrease in what we want to see. So we'll tumble. And that didn't work out well for us. So we will tumble again. Run, and you get this. So it ends up working progressively overall in a direction, how frequently we're going to tumble versus run in order to control our directional movement. But you can also start to perceive here that if you have just one flagellum, you have less control over this. If you have a whole bunch, they can control them individually. And so that would allow them to have a little bit more control over their directionality with their runs and tumbles, with their individual, like all those many flagella everywhere. So yeah. When we start talking about the eukaryotes, they also have flagella, but theirs moves in a whip and they can control it in the whip. And so that allows them to actually control their direction very precisely. So that's a big difference when we're looking at eukaryotes versus these akaryote guys. So these guys just random runs and tumbles based solely on the rotation of their flagella. Let's talk about how they attach to things and other things that they do. So they've got a couple of structures on the outsides that they can use for attachment, and flagella is the only one that allows for movement. I want to keep that clear. These guys are for attachment, the fimbria and the pilus. I'm going to talk about them individually. Start with the fimbriae. This is multiple. Fimbria is single. So the fimbriae, they're like hair-like bristles. They're much shorter than flagella. You can kind of see that in the picture. That's little hair-like spikes, and they're made of protein, and these proteins can attach them to surfaces, and that's really all they're for is for attachment. And so some of these fimbriae that some bacteria have are better at sticking to certain surfaces than to others. And then maybe these guys are better at those than they are at these, and whatever. So there's certain strains of E. coli that have really great attachment that's so effective that they can even attach within your urinary tract, and they don't get flushed out by your urine. Now, E. coli doesn't belong in your urinary tract, okay? It's not part of your normal flora. So not in your urinary tract, but it is part of your normal flora of your gut. Most people, and I mean a great majority, if not close to 100%, will have E. coli as a normal part of their gut flora. And we have a ton of different gut flora, but that would be one of them. And 80 to 90% of urinary tract infections are caused by E. coli from your own butt, okay? And especially for women, and this is why in women we teach you, wiping from front to back instead of the other way, because you don't want to spread your E. coli into your urinary tract and risk a urinary tract infection. And of course, whenever we're engaging in things like sexual activity, there's a lot of mashed things around, and things can get a little bit, maybe introduced into areas where they don't necessarily belong. And, practicing hygiene, urinating after sexual contact and things like that. We'll talk a little bit more about that whenever we get into sexually transmitted infections later on. But that could be important for maintaining the hygiene of those areas. But E. coli has some strains of E. coli have such great, strong fimbriae. They don't care. There's not really going to be a whole lot you can do to urinate these guys out once they establish themselves. Those diffusely adherent E. coli have very effective fimbriae, and that's how they're attaching. So it's very important. 80 to 90% just E. coli. All right, so the pili, they can be used for attachment, whatever, but they're the one that I want your take-home message to be with the pili is that they are used for connecting cells, almost like a channel or a tunnel between cells. And what they use this channel for is exchanging DNA. So they call this conjugation. So you think of a conjugal visit, maybe at like a prison for somebody, and you know what's going on in that conjugal visit, sex, if you didn't know. But we think of sex as being a way of us exchanging DNA, swapping DNA. Well, these bacteria are going to swap DNA, and they'll take like extra bits of DNA that might be things for antibiotic resistance or making certain extra toxins and things like that. And they can make little copies of them and then shuttle them through these little tunnels to the guy next to them. So maybe this one guy randomly acquired this mutation that helps with, antibiotic resistance. It can copy that and shuttle it through these tunnels to the other cells nearby. And now suddenly you've got a real problem with spreading around antibiotic resistance, okay? Only gram-negative bacteria can do this. That has to do with the structure of their cellular envelope. But conjugation happens through pili. Sex pili is what we typically call them. Moving on to that sticky coating we were talking about previously that allows you to make biofilms. This is called the glycocalyx. Calyx means coating. So it's like structure around these cells, coating around them. Glyco, sugar. We always be thinking sugar when we hear that. So it's sugar-based. It can have proteins in it, but more importantly, sugar-based. So think of this as being like almost like a layer of honey, sticky honey around these guys, right? So this can be in the form of a slime layer or a capsule. We'll talk about capsules more commonly than slime layers, but slime layers are just loosely associated, more gooey, if you will, whereas the capsule is a little more densely associated. And a capsule is actually more beneficial for things like pathogens because it doesn't really get in the way of them existing in an environment, by the way that a slime layer can be it can almost impede them creating, illness and things like that. Either way, these are sticky, sugary coatings. We're going to talk about the capsule the most. It's more dense, it's more tightly associated. So here we can see a stain. It's a negative stain. We've got a background going on here. Our positive stain for our cells, and the capsule itself did not stain. With what it is made out of, it's not just sugars, but the other things that are in it, it will not stain well with the stains that we use. So it'll be colorless. And we'll actually do this stain when we're in our own lab. It'll be a little bit different color, but it's very similar. So this halo of non-color, this colorless halo, looks white to us because of the light, is the capsule. And that part in the middle, that's the cell. And what shape is that? Bacillus, right? So anyways, these guys have capsules very clearly. This, you see this pink spot out here? That could be a cell. It would just be a cell that does not have a capsule. That's what one without a capsule would look like. There's just no white around it. It would just be cell color and straight up background. No white. So you can see the difference. And we can actually, some cells will only make capsules under certain conditions. So you could grow them and they don't need a capsule. They might look like that. And then treat them in a way that they want to have a capsule. They'll start making it and they get gooey like this. This would be the same species. It's just whether it makes a capsule or not. So you can see the difference there, even just on the actual plate and how it grows. Now looking at the actual growth and what the growth looks like. And like this has almost like a white color, very, very tiny little dots, right? You can see the tiny, tiny, tiny dots. Not as big as some of the other ones we've seen. So all of these observations that we would make about what that growth looks like on that plate, that's called colony morphology. So morphology, talking about what it looks like to us and the colonies. This is something we can observe with the naked eye. And we'll talk more about that in the lab, but I wanted to introduce that concept because it's something that people get lost on later. So moving on, sticking with our glycocalyx, again, specifically the capsules, the densely associated ones. Obviously, they are useful for sticking to surfaces. So that'll help us stick to things and adhere. They're also pretty useful for creating this biofilm, so building up on other layers of cells. But one of the things that it also helps with is if I'm talking about having a nice sticky coating like of honey on your body, it's going to be easier for you to slip away from things that are trying to grab onto you. And so that actually helps a lot with these guys evading our immune system. So our immune cells that are initially trying to eat up all our bad guys and get our immune system going, our white blood cells, these guys that eat up our bad guys, they're called phagocytes. So phago means eating, cyte is cell, cytocell. Okay. So eating cells are white blood cells that go around eating up the foreign things. They can't grab onto them and eat them very well if they have a capsule. We're going to talk about how our body deals with that in later chapters when we get into immunity. So we have a way to compensate for that. But this is actually why a lot of our pathogens have capsules. It just helps with that. All right. So the cell envelope, moving on to the next thing. Now the cell itself, we've all talked about how cells themselves are going to be able to reproduce copies of themselves, but they all are contained within a membrane. And we've learned about phospholipid bilayers for bacteria and for eukaryotes. So that's our cytoplasmic membrane, the membrane that contains the goo inside of the cell is called the cytoplasmic membrane. The goo, again, cytoplasm. Okay. So the cytoplasmic membrane, the membrane that contains the goo. These guys also, bacteria generally, we're going to talk about one exception, generally have a cell wall. Their cell wall is typically made of something called peptidoglycan, which we'll talk about in a moment in more detail. But that cell wall is going to give structure and support to those cells and help them survive certain environments. So those are the two absolutely necessary components for Again, there's only one exception, but those two are found in almost all bacteria. And then we have a very, very distinct group, a big group of bacteria that may also have an outer membrane. But whatever it is that you have in your bacteria, whether it's just a cytoplasmic membrane or the typical one of cytoplasmic membrane in cell wall and or an outer membrane, all that outer layerage that we got going on, that's our cellular envelope. All right. Our single protective units. And this brings us to the Gram stain. The Gram stain is a differential staining technique that allows us to delineate between gram positive and gram negative bacteria. And quite literally, the way you're defined as gram positive versus gram negative literally just means that you stained positive or you stained negative. You stained purple for positive or red for negative, which makes it easy to remember, I feel like, which one is which. P, purple, and then to me, red is a negative color. Okay, so those are the two colors for the Gram stain, okay? So you're either positive or you're negative. As it turns out, if you're positive, you tend to have a thick cell wall, like a big, big scaffolding cell wall, many, many layers thick, and then you have your membrane underneath it. Whereas the Gram negative staining, the ones that stain red, they tend to have an outer membrane, a very thin, I hate this, I should have used the other thing, a very thin peptidoglycan, and then a cytoplasmic membrane on the inside. So it's like membrane and membrane sandwiching a very thin wall. So I drew this really terribly. Let's show the picture that's better. So here we can actually see that our gram positives, which are going to stain purple with our Gram stain, have this very thick peptidoglycan wall. In fact, it's so thick that we have to have other molecules help us maintain it and prepare it for whenever we're making copies of cells to grow them new thick walls as well. And those molecules that are helping out with that are lipoteichoic and teichoic acid. We'll re-mention these in a moment, but these guys are helping with the structure of that thick cell wall. So this is the makeup of the Gram positive wall in a gist, okay? For the Gram negative cellular envelope, they have this thin, sometimes even a single layer of peptidoglycan. Because they don't have as much of a wall supporting their shape, they're actually going to need that extra membrane on the outside just to help maintain with their shape and everything like that. But now we can sort of start to get that there's quite a drastic difference in their cellular envelopes. Very different, chemically speaking, as far as just going to be functionality. As it turns out, that thick cell wall of the Gram positives tends to lend to a stiffer, stronger structure. And so those guys are, because they attach their membrane on the inside of it, sort of like, think of like a balloon being stuck on like a brick wall around it. So we stick that balloon up on that brick wall, and we remove all the air from the balloon. The balloon shape is still going to hold because it's stuck onto the bricks, right? If we don't have brick, but we have like paper around it, that whole thing might still collapse. So think of it that way. The brick surrounding the balloon that it's attached to versus paper for the balloon. So the paper would be the Gram negatives in this case, right? So have another balloon on the outside might help a little bit, but it's still not going to be anywhere near as strong as the brick. So because of that, we can pop things that are like Gram positives. They're going to handle being put into saltier environments a lot better because the balloon is stuck to the brick wall. Whenever you put it in salt, well, we know if we put cells in salt, they're going to shrivel up, right, and dry out, sort of. So losing all their water. Well, these guys aren't going to shrivel. They might lose their water, but they're going to still maintain their shape. So they can survive a little bit better because they're not losing that. So things that grow on your skin tend to be Gram positive. Your skin's pretty salty. So those guys are typically going to be Gram positive bacteria. Whereas our gut bacteria, not all of them, but a lot of them tend to be Gram negative. And so those guys tend to be better adapted for that environment. It doesn't matter why, but they just do, okay? But that saltiness, now you can start to see what the importance would be of the stability. It gives it more stability, more strength, more structure, but it's not always favorable for every environment, okay? So the Gram negatives, they are a little bit more flexible, more susceptible to being affected by salt and stuff. But that flexibility gives them some advantage in certain environments as well. In the Gram negatives are red staining guys. They also have on their outer membrane. So you see we have an inner membrane here. We have our peptidoglycan sandwiched in the middle, very thin layer, not brick walls. So we have paper here. and then an outer membrane on the outside here. It's also just a lipid bilayer. Just regular membranes here, okay? But on the outside of the gram-negative, we have some special molecules as well. So while our gram-positive had our techoic type stuff helping out with the structure of that, our gram-negatives, they have this special molecule on the outside called lipopolysaccharide. Another molecule that is going to tell you kind of what it is. So lipo, we've got lipids involved, hydrophobic regions, and then we have the saccharides. So we have sugars and fats, right? And so that's what that molecule is made out of. We also call this LPS. And then that LPS, we have, this is like our chemical wording for it. It's what sciencey people would call it. But in medicine, they have their own terminology for this. And this molecule in medicine is called endotoxin. An endotoxin, excuse this wonderful handwriting, endotoxin, LPS, lipopolysaccharides, all the same thing, okay? And I'm gonna talk about it a lot in the semester. But endotoxin, actually, your body sees it immediately, and it will react because it's not supposed to be there. Like if it's in your bloodstream, for example, it's definitely not supposed to be there. Again, blood shouldn't have bacteria or anything in it. If it does, you're infected. So if we see endotoxin in your bloodstream, your body knows instantly we're making a fever, we're starting the inflammatory cycle. But the inflammation can be very damaging to your body as well. So it can lead to shock, very low blood pressure, very suddenly, and even death eventually if it's not, maintained properly. So endotoxin is one of those things. Endo, inside is what endo means. So it's inside because it's part of those gram-negative cells. They're not secreting a toxin. It's actually part of them. And so there's the endo part, and it's toxic to us because it will lead to damage to our cells. So that's a really important part medically that we're going to bring up a lot as we learn more about it and how it leads to this damage and everything like that. Okay. So endotoxin is really important. It's only found on our gram-negatives. All right. So peptidoglycan, it also tells you what it is in the name. So we already said when we're talking about glycocalyx, that's sugary coating. So glyca here, that's going to be sugar. Peptido, we're literally talking about peptides here. You guys remember what peptides are? Short chains of amino acids, 20 or less, right? So what's going on with peptidoglycan? And you don't need to be able to draw this or recognize it or tell me what the M's or the G's stand for, none of that. What I want you to know, actually, let me get rid of this, whatever this is. Hold on really quick. I want to just go to not this. And then I want, did this stop recording? Hold on really quick. I've been zooming, right? You gotta love it. It's still recording, it just isn't showing the right thing. All right, I don't know. Anyways, so this will be better because I can choose the colors faster. So we will have these chains of the sugars, and this won't do that weird line thing that the other thing does. Chains of sugars, okay? It's just two sugars, and that's what the G's and the M's represent. Just, for the sake of science. What the M is N-acetylmuramic acid. I'm never going to test you on it, okay? But that's what it is. And the G is N-acetyl glucosamine. That's what it is, glucosamine. N-acetylglucosamine. So that's the G. So it's alternating N's and G's and M's and G's and M's and G's, just alternating. That's what this represents in the yellow up here, the gold. Okay, I'm so sorry that I wasn't showing. I had no idea. It didn't tell me. Okay, well, it's up now. And if you need to go back and look at the actual images in the first part of the lecture, they are on the other recordings. Unfortunately, it's the best I have for you. I apologize. So anyways, these green lines here represent our peptide cross linkages. And so you can see here, we're starting to build almost like a scaffold. And it is quite like a scaffold. So we have layers and layers of these sugar chains, and then in between cross-linked by peptides. Make sense? So it's important to understand that this is built like this because of how the gram stain works. And we have to actually understand how the gram stain works. I'm sorry to break it to you, okay? But that's part of this. So this is down here, it's just showing you that it's amino acids linked between each of these sugar molecules. Cool? Scaffolding is what you need to take home from this. Sugar chains linked across by peptides, short chains of amino acids. That's peptidoglycan. All right, this is part of the sex about this. next. All right. So we already talked about teichoic and lipoteichoic acid. They're helpful for maintaining the cell wall if there's damage, helping repair it, as well as helping build new ones in new cells as we're making them. They also give that acidic charge. They maintain that acidic charge on the outside, that negative charge that we would see from the membranes themselves. So we're still maintaining that. Very thick layers of this. So 20 to 80 nanometers thick. So 20 to 80 versus the 1 to 3 of the peptidoglycan that we see in the gram negatives. It's very thin. So we're talking like anywhere from like, you know, 20, a good 20 to, well, I guess 80, but as much as 80 times thicker on that peptidoglycan. So it's much, much thicker for our gram positives versus our gram negatives. We still get a little bit of structure from that cell wall, but we do need a little help. And that's what that outer membrane is going to be doing for those guys. And again, the lipopolysaccharide is key here. This is it showing us that lipopolysaccharide, again, sugars. We see that depicted by these nice happy hexagons up here. And then this down here, these chains represent carbohydrogen chains that are going to be, you guys remember the word for it, right? Hydrophobic. All right. So not everything is going to fall into those boxes. Of course they're not. So we've got two exceptions, all right? Now this guy's still going to be having peptidoglycan wall, but it's just slightly different. Normal peptidoglycan wall, you have that thick wall, it's gram positive, and then you have the membrane underneath. This guy has a partial situation here, but instead of being full peptidoglycan, on the outside, it has these waxes. Think of these as hydrophobic waxes that are kind of attached within that cell wall, okay? And this wax is called mycolic acid. It's like a fatty acid. So it's very, very long fatty acid chains that are waxy. And they give a waxy coating on the outside. So now we're talking about the outside of the cell not having that acid component, but being hydrophobic. That actually is protected for these organisms. They're not going to dry out as easily. And they're going to be very resistant to chemicals and drying out in the environment. In fact, these bacteria can like hang out in the air and in the environment for like hours after being released into the air. So 8 hours after somebody coughs in here with one of these bacteria in their lungs, it could still be hanging out, perfectly alive, ready to infect somebody. And the bacteria that affect your lungs that have this on their surface, those guys, they are they're only take 10 of them to cause an infection on average. So viruses, you only really need one virus. The bacteria sometimes need a little bit more than one to get colonies and get them interacting and stuff like that. So 10 is pretty low for bacteria. To put it into perspective, salmonella, you need at least 50. So 10 is pretty low. So anyways, so these are mycobacterium. So they have myco in the name, and there's nocardia. I will literally never test you about nocardia. I'm telling you that it existed. Never going to test you about it. I said it, okay? But you absolutely need to know mycobacterium. And it's in the name, myco, has mycolic acid. That's the wax. Two organisms that apply to medicine that you do need to know. There's more that are affecting medicine than just these two, but these are two biggies. Mycobacterium tuberculosis, which causes tuberculosis, or Mycobacterium leprae, which causes leprosy. We don't see as much of that, but it is something that people are aware of, right? So you might not think we don't see a lot of tuberculosis, but like I've mentioned before, 1/3 of the world carries tuberculosis. 1/3 of the world carries tuberculosis. That is no small number. So this is very medically relevant. We do have quite a lot of it here in the United States, just not typically in our healthier populations, though I did mention previously, we had an outbreak at Edmond schools just a few years back. Healthy people, students, No, of tuberculosis. Yeah, not of leprosy. Yeah, leprosy is not actually too easy to contract. It's kind of harder to get that one, despite its sort of reputation. But anyways, these guys are very slow growing and it's really hard to culture them. Luckily, there's a really easy way to tell if they're there, and the acid-fast stain does that for us. The acid-fast stain uses a lipid-soluble stain that will dissolve and stick to these lipids up here, these waxes, that won't stick to regular cells that don't have that wax. And so you can have your patient cough into a cup and send that sputum up to the lab, and they can literally stain the sputum without having to culture it and see if there's acid-fast bacteria in there. If there are, they have to be mycobacterium. The only bacteria that are medically relevant that are acid fast are Mycobacterium. They also always have that name, Mycobacterium blah, blah, blah. Okay, so that's Mycobacterium. The next other non-typical one is going to be also, so I want you to remember Mycobacterium, okay? If you've been paying attention, you looked ahead, then you know what's coming next. Mycoplasmas, all right? These guys, I do not want you to focus on the myco for this one. What I want you to focus on these ones for is the plasma. So it's either mycoplasma or mycobacterium. So if it has plasma, what I want you to think about is if I'm walking around, if this was like gross, but like if it was somebody's literal plasma, the liquid portion of your blood, that golden yellow liquid portion is mostly water. If I was carrying that around in this cup and I were to spill it out on this table here, would it stay and hold a shape every time, a defined shape? It just go like water, do whatever. So it's an unpredictable shape. Same with these guys. They're pleomorphic. Why are they pleomorphic? They don't have a cell wall at all, not even a thin one. So these guys will stain gram negative because they don't have a cell wall. We'll talk about how the stain works and why that is. They're staining gram negative, but that's because they don't have a cell wall at all. And they can't maintain the shapes of their cells. They'll have sterols like we have in our cells, but that doesn't give them enough structure to maintain shapes. So they are pleomorphic, like have random shapes. All right, so that's how I remember the difference. When you hear mycoplasma, focus on the plasma. Okay, you hear mycobacterium, it's bacteria. Mycoplasma, pneumonia. Yes, so that's walking pneumonia. Yeah. Yes, it's pretty, that one's a pretty common one right now as well. It's going around. Good call. All right. So the whole point of the cytoplasmic membrane is to keep what's in the cell and keep what's out of the cell out of the cell. It has selective permeability. Only certain molecules can just cross the membrane freely. Very few, water and a few uncharged molecules. And that's how we control what stays in and what goes out and things like that. If we're trying to move molecules on our own and control that, we'll need to have proteins that help with that. And that's how we'll be able to secrete or discharge things out of the cell, like waste or toxins or things like that. So that's kind of the purpose of that. So how does the gram stain work and how's that going to apply to everything we just learned about the whole cell envelope? All right, let's buckle down, folks. Are we ready? Crystal violet is our first step. Now it tells you what color it is, right? Purple. It's basically just a stain, and it'll pretty much color anything purple temporarily. So we'll add that first. What happens is the crystal violet, and I've already told you, we're going to have layer upon layer upon layer of this scaffolding. And it's sort of like a Plinko machine. It's going to trap the crystal violet molecules in that scaffolding. The bigger the scaffolding, the more molecules you're going to trap. So the gram positives, those thick scaffoldings of peptidoglycan, they'll trap a ton of crystal violet. The gram negatives, they'll hold on to it temporarily, but it won't stay when we start to rinsing it with alcohol. Okay, so we'll get to that. So first we're going to fix things in place with iodine. So Graham's iodine is something called a mordant. So think of like, I feel like it sounds a lot like mortar, like brick mortar. This is a mordant, and that helps fix the dye into place if it gets trapped in there appropriately. So we're not fixing anything on our gram negative because it's not actually stuck in there really. It's just kind of just chilling. So we can't get cross-linking going on with our iodine there because there's not enough of them that are really staying and getting caught appropriately. But when we rinse it with alcohol, alcohol for us in this particular situation is the decolorizer. What this is going to do is remove unfixed crystal violet. So if it didn't get fixed by the iodine, the alcohol will decolorize. And so we can actually see that if we were to look at it in our microscope once we got to that step, that gram negatives aren't going to be fixed in place with their crystal violet. We would rinse it out at this point. So now our cells are colorless if they're gram negative. So we can't see that in a microscope. I hate to break it to you, but it's like impossible to see unstained cells. So we will counter stain it with our safranin. So this is our counter stain, so it'll help us see our decolorized cells. So counter stains help see decolorized cells. And those are typically our negative ones. Decolorized or negative cells. All right, so that's what our counter stains are going to be doing for us in this situation. So this is where we're getting our purple versus our red. Make sense? conceptually. And like I said, we're going to be doing this in exercise 3. So that will be on our review day once we get to that. So yeah, so you'll get to do this before we get the test done. So get this in practice and get to see how it actually works, and that'll help with understanding as well. So back to what's inside of our cells, the gel where everything's existing is our cytoplasm. It's got all of your ribosomes and everything that's in there, our DNA, and then all the sugars and proteins and amino acids and all that just chilling in there, right? DNA for our bacteria, they do have DNA as their genes, but it's going to exist in one big circle, just one big circle. We have 23 pairs of linear chromosomes. These guys have one big circle, and it's pretty regular for bacteria to have that. And so this isn't contained in a nucleus. It's just kind of hanging out in an area called the nucleoid region. They can also have extra little bits of extra genes on their DNA that are contained in these things called plasmids. Plasmids are also circle bits of DNA. They're very small circle bits of DNA, but they'll contain things like antibiotic resistance or extra toxins that they can make or enzymes to break down other things. So that can be extra information that can help them survive, but what information is on a plasmid is never essential. So the cell can actually exist without it, but these things might help them exist more easily. And it says they carry traits that will help with, it says protective traits, something like that, helps with something called pathogenicity. And that's just a term that refers to their ability to cause disease. Ribosomes. These are made of RNA typically, and a little protein, but the RNA is what's key. The RNA machines that are going to help us make protein later on. We'll learn about that in chapter 6, so don't focus on understanding it really beyond that right now. All right. For bacteria, their size is denoted as 70 Svedberg units. I will never test you on a Svedberg unit or that it's 70 versus 80. But what you should probably be aware of is that bacteria have their size of a ribosome, and then eukaryotes have their size of a ribosome. Archaea have the same size ribosomes as eukaryotes, not bacteria. And that's something. And I'm also going to tell you this. Your mitochondria have their own ribosomes, and guess what size they are? Bacteria-sized. They also have their own genomes, circular genomes. They divide on their own. Your genes do not encode anything for your mitochondria. So we're starting to see a little bit why people say mitochondria may have just been bacteria that we absorbed that eventually became something that we were dependent on. Pretty cool stuff. So we'll talk about that when we get to the eukaryote chapter. Now we're getting into endospores. We're almost done. Endospores are structures that bacteria can make to withstand hostile conditions. So it's like taking the cell, condensing it down to just the stuff that you would need to revive yourself. That's it. And so this is not a reproductive thing. One cell will lead to one endospore, and only genera Bacillus and Clostridium can make these. So you guys remember how we talked about acid-fast bacteria having mycolic acid. Those are always Mycobacterium. So their name is literally going to be Mycobacterium something. You talk about mycoplasmas not having a cell wall. Those mycoplasmas, like Mycoplasma pneumoniae, they always are going to be called mycoplasma something. So now our endospore forming bacteria are going to be called Bacillus something or Clostridium something. As it turns out, Clostridium bacteria, all of them in that genus, they all hate oxygen and they don't like oxygen and they don't survive well in oxygen. But what they can do is hunker down and wait till they're out of oxygen. Like once they're in a non-oxygen environment, they can thrive. They thrive pretty well in your gut, for example. This is what causes C. diff. What do you think the C stands for? Clostridium difficile. So these guys get out of someone's booty and they can hang out in the oxygen as endospores and just lie in wait until they get into somebody else's colon, but through ingestion, typically through poor hygiene practices. And those endospores are really hard to kill. These guys are super hardy. They survive boiling. They survive boiling, y'all. All right. And a whole bunch of other things. Autoclave will kill them. We'll talk about how and what that is and everything. It's okay if you don't know yet. But there are things that can kill these guys, but they are very hardy. Okay. So these guys are obviously, if we're talking about C. diff, there are species that fall within these genera that can be infection causing, quite serious. And the fact that they can make endospores defines their ability to cause these diseases that they do. When they are multiplying and they are making toxins and they are making new cells and they're metabolically active and all these things, Those are vegetative cells. In fact, any cell, whether it can make an endospore or not, whether it's capable of or not, even if it's not Bacillus or Clostridium, we're talking about E. coli or staph. If it's active and multiplying, it's vegetative, okay? When Bacillus and Clostridium go into lockdown mode, that's the endospore. The endospore is completely dormant. It's not making toxins, it's not multiplying, it's literally just waiting to become a vegetative cell again, and then that can multiply and divide and make toxins and things, okay? So endospores are completely inert and dormant, and they're waiting to be woken up again by a nutrient or water or something like that that revives them. The process of making an endospore from a vegetative cell, going from vegetative to endospore, that's called sporulation. Again, they can resist heat to a certain point, drying, almost completely desiccated. We can completely desiccate some of these guys and turn them into a powder, and then use that and spread it around to people, and then cause that as a bioterrorism agent. Do you guys have an example of what you think of that we already do that with that exists like that? Anthrax, maybe? Okay, Bacillus anthracis. That's what that is. So you're learning things. All right, so they can resist freezing, radiation, and a lot of different chemicals as well. All right, so very resistant. Once they start seeing water or nutrients or something like that, they can start to revive. The cell that's making the endospore is called the sporangium. It's the sporulating cell. So remember, sporulation, making the endospore. The cell that's making it is called the sporangium. And that process takes about just 6 to 8 hours to go from vegetative, shrink down, pare it down, give your nice cortex coating to protect your DNA inside and everything. 6 to 8 hours. So this is the process of how that would work. It's all laid out. It's stepwise explanation of everything, but you start with a vegetative, happy dividing cell. You go through sporulation, through this process here where you make your spore. Your spore now, as sporangium has made your spore and released it, it'll just chill and wait until it gets the signal to germinate. Then it goes through germination to go back into the vegetative cell. As it does that, germination, exposure to water or nutrients or whatever, it only takes about an hour and a half to become vegetative again. That is impressive. So think about this. These are some of the guys that are endospore forming that are related to disease. There's more than this, but these are some examples. Bacillus anthracis, anthrax. We've already talked about how you can dry it down. So they attacked Congress back in 2002. Literally, somebody mailed them anthrax, and that's what happened. Clostridium tetani, this is tetanus. This causes not just lockjaw. A lot of people know it as lockjaw. Easy death from this, no joke. It's why we vaccinate, and it's why your doctor, you go step on something and get a pokey, and they're gonna be like, when was your last tetanus shot? Oh, it was about 5 years ago, you're good. Every 10 years, you should be getting a booster for this. And your doctor will usually keep up on that if you get any serious injuries. But these guys have to get pretty deep in there to get out of the oxygen and then turn back into vegetative again. But you step on a nail deep enough in there, these guys can just be hanging out in your soil, just in your backyard. In fact, botulinum, Clostridium botulinum, that leads to botulism, it's like everywhere. And if you're brewing beer, you need to be sterilizing, not just boiling your equipment, but properly sterilizing with proper chemicals and things like that to eliminate the likelihood of Clostridium botulinum getting in there. So Clostridium tetani leads to lockjaw. We're talking about seizing up of your muscles to the point you cannot relax them. You lock your jaw in place like that, but eventually even your diaphragm will get locked in place and you can't breathe and you will die of suffocation. With Clostridium botulinum or botulism, most of us know this relaxes your muscles. So these make it so you can't contract your muscles. So this is flaccid paralysis now. Spastic paralysis versus flaccid, but you can see how they can be related to one another. All right, C. perfringens, Clostridium perfringens. This is gas gangrene, another one that's just like pretty much everywhere. If it gets deep enough in your body, you get gangrene, okay? Archaea live in extreme habitats. That's pretty much what to know about them. They can make things like methane gas and can live in like the colon of you or cattle or whatever, and they're farting out methane, but it's the bacteria that are making the methane. Methane's a worse contributor to global warming than carbon dioxide. They can also live in extreme salt conditions. So halo is referring to halogens on the chemical periodic table of elements. Halogens like chlorine and things like that. So these guys can exist in very high salt concentrations that most things would die, and they can actually live in salt lakes and salt mines and salted fish and all sorts of stuff. They can also live at temperature extremes. If it's really, really cold, we call them psychrophiles. If it's very, very warm temperatures, those are hyperthermophiles. Phile being love, so they like whatever. Psychro apparently means cold. So the Hyperthermophiles can flourish, between the temperatures of 80 to 113 degrees Celsius. Boiling is 100 degrees Celsius. I want to be very clear about that. Boiling, so we're talking about flourishing in temperatures above boiling. Very impressive, right? So those are the guys that are like in the deep sea vents. And then we can actually break down and classify Archaea versus these kinds of different groups of bacteria based on what kind of cellular envelope they have. So we have 4 ways of classifying them, and this is how I remember them. All right, so I always start off with the firmicutes because it's the easiest for me. Because firm, I remember very easily that's the thick peptidoglycan firm wall. So that's our gram positive. Gram positive, they have the thick walls. Next, I'm like, okay, we took care of gram positive. Which one's the gram negative? The GRA, the next one, gram, GRA, okay, that's how I remember it. Gram negative. Taking care of gram positive and gram negative already. My next one that I remember is Joey Mendoza. I already explained this to you guys. But archaic, that's a long time ago, so old or whatever you want to think about it. And not a long time ago, I thought Joey Mendoza was cute. So the Archaea fall into the Mendoza cutes. That's how I remember that literally. And then we have tenericutes. Tenere in Latin means it's the same thing essentially as tender. So think of tenor as being just a misspelled tender. They're very tender because they're very soft because they completely lack cell walls. Okay, so that's how I remember which one falls into which category. Last thing, Berge's manual is a book that can contain E. coli has lophotrichus and it is gram negative and it has all these traits about it. And you can look these up in a manual and compare traits of your bacteria to figure out, oh, I found a bacteria that I see that is lophotrichus, that is gram negative. What could it be? And look up those traits and figure out what it is. There's a whole manual for this. It's massive. It's multiple books in its physical form. It's called Berge's manual. You just need to know what it is. Okay, so it's used for identifying these guys. We can actually identify them based on the species. So obviously E. coli versus Staphylococcus aureus are different species. But we also know that E. coli that lives in your gut normally is completely different from the E. coli that you can get from contaminated food that they send out the warnings about. That can't possibly be the same thing, or they'd be worried about the E. coli in your gut. So the E. coli that they're worried about is a strain called O157H7. And O157H7 carries a special gene that is from, it shares it with another organism called Shigella. It's called Shiga toxin. And what that does is it breaks down the lining in the cells in your intestines and leads to bleeding. So you get bloody diarrhea. The word for bloody diarrhea is a word you've probably heard if you've ever played the game Oregon Trail. It's called dysentery. So dysentery is the word for bloody diarrhea, okay? And so if you have the strain of E. coli that causes dysentery, it's a different strain than the normal strain in your gut. So if it just has an extra trait or something, we'll call it a different strain. If it has a different surface marker than another one, then we call it a serotype. We'll talk about why that is in later chapters, but that's it. So real quick, our questions: round cell shape, cockeye, right? Appendages that we use to move, flagella. Chemotaxis, responding to chemicals in your environment and moving towards or away from them from that, right? And they rotate their flagella to do that. Runs and tumbles, right? So gram positive versus gram negative. The big traits, the gram positive, thick cell wall. The gram negative, thin cell wall, extra membrane on the outside, okay? Then we have the endospore. That's our protective structure. It's completely dormant. It is not metabolically active. It's just waiting for favorable conditions, and that's it. So we'll head up to the lab now. Guys, take 5 minutes, go take your break if you need one, and then once everybody's up there, we'll go ahead and get started. Please don't take longer than 10 minutes so we can get out of there early. As soon as everybody's up there, the sooner we get out of there.